Actually, we're going to get started here with just a little bit of an overview. I think Tom from CDC wants to just outline the problem as you see it and some of the ideas in the pipeline.
Great. Well, thanks. No, thanks for having me. I'm Tom Chiller. I'm the chief of the Mycotic Diseases branch at the CDC in Atlanta, Georgia. And so I work on fungus 24/7, as we like to say. And
glad to hear it.
I think that there are definite challenges in the world of fungus. And fungal infections, I always like to refer back to Rodney Dangerfield as getting no respect. I think oftentimes we forget about fungal infections. We don't want to hear about fungal infections, and we don't, unfortunately, give them the same respect and credence that they deserve in taking care of our patients. And so, you know, I'm an infectious disease doctor by training. I've been treating patients with bacterial sepsis for many years, but also, unfortunately, been treating patients with fungal sepsis. And in fact, in today's world of sepsis and bloodstream infections, Candida and candidemia in many places is the #1 cause of bloodstream infections. So we've done a great job actually of beating down bacteria, but we haven't done the same job beating down fungus, and so fungal infections continue to remain, you know, a major concern and a problem. And then I've been at CDC now almost 20 years, and honestly, I talk about fungal infections. I'm here trying to make people aware and to hear about them. But I never really thought I would see what we've seen emerge over the last several years, and that is, in 2009, we began hearing reports about a fungus, a yeast called Candida auris. It was a new species. It was isolated from the ear, therefore auris. And then we began hearing that this infection was in the blood, that it was causing candidemia, it was causing bloodstream infections, it was killing people. Potentially, it was in India and South Africa and Pakistan and Venezuela, and Japan and Korea, and we started paying a little bit more attention when colleagues called us from Pakistan and they reported a cluster of infections in their hospital that seemingly was being transmitted person to person. They thought it was caused by the fungus Saccharomyces, and it turns out it was actually the fungus Candida auris, because Candida auris is incredibly hard to identify biochemically using the normal and regular techniques. Fast forward now to where we are today in the United States, and we have now over 100 cases of this organism in the US. We know this is a fungus acting like a bacteria. You have to think of it like a Acinetobacter or MRSA. It's transmitted person to person readily. It grows on skin. It loves growing on skin, actually. It grows in high salt, high heat. You can culture it weeks to months from a patient's skin after they've actually had the infection. You can culture it on surfaces, it's on hospital beds, it's on windowsills, and worst case, it can acquire resistance to all 3 known classes of antifungal agents. So it can become the true superbug of the fungal world that I never really thought I would see, at least I was hoping I never would see, and now I'm seeing it being transmitted like a bacteria. And so even though I'm out here and this panel will talk today about progress being made in, you know, with fungal infections, here we have a fungus that's acting like a bacteria, and that's creating a whole new set of challenges in communication about these organisms.
Now, just one real quick one. You said it's hard to identify biochemically. Can we now better detect it with DNA sequencing that's around?
So yes, sequencing detects it, and the 2 machines, well, the main technology out there now is MALDI-TOF. You might have heard of MALDI-TOF. MALDI-TOF is made by 2 different companies, and both of those companies' machines can detect it, but not all of those companies' machines. So they're working on upgrading their software. The bottom line is all the other ways we test for Candida do not detect this species. They will find other species. It'll get to a road where it says unknown species, or it'll get to a road where it actually identifies it as something else. But that becomes challenging, because then we work with institutions. About 1/3 of the clinics around the country in the U.S. have MALDI capability. So it's still lacking. We're still not identifying it, and that makes it challenging to react to.
So 100 confirmed cases, but no likelihood, many more out there that we
Could be
just
many more.
haven't yet seen. Okay. All right. Well, thank you very much for that helpful kind of starting point, Tom. I think, I'm not the expert. I'm the journalist up here. I think if I do know something, it is about the perception part, where you were talking about the Rodney Dangerfield aspect of this. It's sort of, I think of it as almost like there's an ick factor around fungus. People don't really like to talk about it. I think if you were to ask most people in the audience, like word association, well, how many people, if I say like fungus, are people thinking, what's the first part of the body that you think of? Anybody want to just shout that? I would imagine toenails, right? I mean, toenail fungus, and it's kind of gross. But that sort of trivializes what we're talking about here. I don't think anybody up here is that concerned about those sorts of things. There are companies out there that do make businesses out of reformulated versions of something or other and raising the price for treating toenail fungus. But this is much more serious. And I was telling Jeff Stein before getting started that one of my experiences as a journalist was I think indicative of the lack of awareness that we see out there and how this sneaks up on people. And just very quickly, the story was with gene therapy. So I was covering a company 10 years ago called Targeted Genetics, and they were working on viral vector gene therapy. They were delivering the TNF-alpha protein directly to treat rheumatoid arthritis in the knees. And they had a patient die in a clinical trial. And immediately, the market, many scientists looked at this and said, aha, here we go again, viral vector gene therapy, it's unsafe, it must be something about that virus. But nobody really knew. It was a medical detective case. This is often the case when something bad happens in a trial. Months later, they find out that this patient had been, well, of course she was immunocompromised. She was on immune suppressants for her rheumatoid arthritis. In addition to this, experimental therapy, but where she lived in southern Illinois, there was a lot of certain species of fungus, and I don't remember which one, but this was known as a pathogen. And when you are immune suppressed, this was clearly like a risk factor. They found out that was what killed her, and so if they had known that earlier, maybe there would have been a way to save her. But the point is, there are now emerging, as you point to this one of Candida auris. There are probably more. A similar story playing out here that we have seen with antibiotics. So in terms of resistance developing over time, emergent species. So I think that's what our panel here is here to talk about today. The serious threats, not the trivial, and we're going to get past that ick factor thing. So who wants to start? Marco, would you like to go for it? Introduce yourself briefly and say a little bit about what your company does, your bite on this problem.
Certainly. My name is Marco Taglietti. I'm the president and CEO of SCYNEXIS. We are a company actually developing a novel antifungal. But let me answer your first question. When someone tells me fungus, first thing I think are truffles and wild mushrooms. But this is probably because I am Italian. I'm an infectious disease specialist by training, and I've been always, of course, fascinated by any kind of infection, especially fungi. And I must say that working for SCYNEXIS has been sort of a dream coming true. The possibility of working on a novel class of antifungals that will allow us actually to treat infection, including actually Candida auris. We have some very positive news recently showing that our product has activity against Candida auris. So I think, I don't want to take too much time as an introduction, but without further ado, saying that I think that certainly there is a great need to develop novel antifungals.
Yeah, thanks, Marco. Bob Schotzinger, founder and CEO of Viamet. Full disclosure, we actually have a program in toenail fungus and recurrent vulvovaginal candidiasis, which we may touch on later.
Make some money there and then put it into
exactly.
this program.
And that's the thought process. But But we have many programs behind that that are more focused on invasive disease, and hence I think the focus today. We got into the antifungal space through our platform. We focus on metalloenzymes and have a chemistry platform to build what we believe are both very highly potent and highly selective inhibitors. And it turns out that the target of the azole antifungals, which are still the most commonly used agents today, is a metalloenzyme in the fungus. So we sort of got into this space through our platform, but I think, have multiple products moving forward in development, and certainly happy to give you my thoughts today.
Great.
Yeah, Tim Cook from Novadigm Therapeutics. We're working on vaccines and immunotherapies against Candida as well as Staph aureus. Our company was founded UCLA Research. It's Jack Edwards has got a great team in antifungals, and they discovered antigens on the surface of Candida, and we've made them recombinantly like a kind of a classic modern vaccine, subunit vaccine. And we've shown efficacy in women with recurrent vulvovaginal candidiasis, which are chronic yeast infections. And it was the first demonstration of an antifungal vaccine efficacy. So it's very exciting for the field and for us. So that's our take on it.
Great.
Great. Jeff Stein, CEO of Cidara Therapeutics here in San Diego. We have a lead program that is completing a phase 2 clinical trial in candidemia and invasive candidiasis. So this is a disease that kills some 67,500 people in the US alone every year. And these are patients that are hospitalized on the best standard of care. So it's kind of a testimony to the fact there is a dramatic need for newer and effective therapies. So why aren't these patients being cured? Well, one of the reasons is delayed treatment. So fungi, as we've heard, are difficult to diagnose. They're not as prevalent as bacterial infections. So typically, if a patient is presenting, and these are patients that are cancer patients, they're transplant patients, they're immune compromised, they present with signs of an infection, immediately the suspicion is a bacterial infection, and they're provided antibacterial therapy. 24 hours later, if a bacteria isn't cultured, then there's a suspicion of a fungal therapy. The problem is the evidence, the data shows that delaying treatment, effective treatment by 24 hours increases your chance, your odds of dying from a fungal infection 30-fold. So that's an important number, right? So it emphasizes the point of getting effective exposure to effective treatment right up front. And that's why some of these rapid diagnostics are really important. But I wanted to just address one of the points that Luke pointed out about the perception and why, here's, you have these diseases of really high mortality. Why isn't getting more attention? Well, number one is, one of the points I mentioned is that these are patients that are in the hospital for something that's much more high profile, right? You're getting a stem cell transplant or a liver transplant, or you're getting some type of fancy immunotherapy. And ultimately, you may die of a fungal infection, but that's not what gets the attention. You died because of cancer or complications due to a transplant. So that kind of gets lost in the shuffle. Also, this general lack of education about how severe these infections can be. And a great example of that is that 3 members here and 1 future member of the antibiotics or antimicrobial working group. It's a coalition of 13 companies that works to drive policy forward to help expedite the development and approval of novel antimicrobials. So we have 4 companies in the antifungal area. We had a meeting with Senate staffers, over the last couple of years, about 6 meetings. And in one of those meetings, with respect to the Disarm Act, which we can touch on later, but it's an act that should be passed later this year, attached to either PDUFA or to the tax bill, that will change reimbursement for antifungals, antibacterials that have QIDP status. Well, the version of that bill that came from the House of Representatives had antifungals in it. Then it went through the Senate committee, and antifungals were omitted from it. So, well, why is that? And so we visited one of the senator's offices as part of he was on the Senate Finance Committee. And we talked to the staffers and said, well, what's going on? Why did you drop antifungals? Turns out one staffer thought, well, isn't that like athlete's foot? You know, why should we include that?
This is why perception counts.
Exactly. It's exactly. And so we had with us Dr. Andrew Shore of George Washington University, who read the Riot Act, says, I have more patients dying of fungal infections than bacterial infections. You know, you idiots, what are you doing? So anyway, they added it back. But it's a demonstration of, number one, you know, the lack of understanding of the severity of these infections. And number two, the influence that a coalition of individuals can have when they help educate and drive policy forward.
I want to come back to the policy because this is important. As I understand it, the GAIN Act, which passed Congress 5 years ago, 2012, which generating antibiotic incentives now, is that the acronym what it stands for? So, I mean, basically trying to address a problem here, which was what we economists would call a classic market failure. And that is we know from public health authorities that we have a problem with antibiotic resistance. And yet the pharmaceutical industry, which has a long, proud history of developing antibiotics, had, for business reasons, largely retreated from the field. They didn't see the kind of profits there that they saw in some other therapeutic categories. They got out. Congress reacted and said, you know, maybe we need to create some new kind of incentives. And that, you know, there's a whole bunch, and Jeff, you're the expert on this. You were part of the working group, right, that helped put that together. So, but there's a parallel here with antifungals. The antifungals got folded into this, which not that many people realize again, right?
Right. Yeah, so you know, the GAIN Act enables companies working on new products that address an unmet need, some significant unmet need, meaning multi-drug resistant bacterial pathogens or fungal pathogens. You get what's called QIDP status, Qualified Infectious Disease Product status, which provides under the GAIN Act a number of important benefits. You know, one of those is an additional 5 years of data exclusivity. So you can have a drug that is about to go off patent, which, you know, from a commercial perspective isn't that exciting to investors to fund your company. But with the additional 5 years of data exclusivity, that can be attractive. It also provides for fast track and the opportunity for orphan drug status. So a number of important benefits to getting that QIDP status. And there are several members of this antimicrobial working group that wouldn't exist if not for the GAIN Act. But you know, I think everybody understands that that's just a great start, but there needs to be some other really important incentives to foster innovation and to foster investment in these companies because of this market failure you speak of.
Now, one of those would namely be pricing, right? Because this is part of the issue. A GAIN Act could provide new incentives and you could get through the FDA, but if the payers say, well, we're just not going to pay much for that drug, you're not able to reward your investors.
That's right.
Are you seeing any movement, and maybe you guys have any opinion there on is there any willingness in the payer community to step up a little more for the antibiotics or for the antifungals?
Well, the issue is, first of all, the GAIN Act certainly created what you can call sort of a renaissance of antibiotics. And we, you know, after the GAIN Act passed, you know, in a few years, we got 6-8 antibiotics approved. It is more than in the previous 10 years. So certainly it achieved the result of having new antibiotics coming to the patients, to the market. However, more than the price, but the price actually at the end, thinking there is a product, for example, I developed in my previous life, Avicaz, in which the pricing has been paid, the payers pay for it. The issue is that the product, given its nature, which is for, you know, severe infection, selected infections, tends to be used in a very limited group of patients. So even if the price is, I would say, relatively high, or for not even comparable to oncology drugs, so and that is a question that we can have some more discussion. Why oncology drugs, you know, you can charge, you know, tens of thousands of dollars, and for a drug that actually saves lives, you can't, like an antibiotic. But that is another story. So the problem is that the population is rather limited, and therefore, with Jeff, I'm part of the same group. We are trying to see if there are ways actually to create incentives that will make companies and investors willing to put money into the anti-infective field, because this is what we are seeing also. A certain amount of reluctance, of lukewarmness now of investors putting money into the biotech companies that are doing anti-infectious drugs.
You say anti-infectives. Is that the preferred term when you meet with investors? Do you say antifungals and they're like,
well,
falling asleep?
Well, it's actually, it's because the issue is for both. Now, I think personally, and will be interested, I think for antifungals, still I believe that it's still a big business opportunity, because antifungals compared to antimicrobials, it's not a crowded market. There are probably 8, 9 antifungals, and in 3 different classes. Well, antibacterials, you have dozens of classes and hundreds of products. So
8 or 9 in development?
No, 8 or 9 on the market. On the market.
Currently
in
available
3 classes.
generic antifungals.
Not all generics, some branded and some generics, but still no more than 8 or 9 antifungals for severe invasive fungal infections. Basically, you have 1 polyene, you have 5 or 6 azoles, and 3 echinocandins. So it's a very small number of products, and so this creates actually both the issue that if one of these classes becomes ineffective because of resistance, there are no options left. And that is a major problem. The second, but at the same time, creates a business opportunity, and this is what I tell to our investors, that there is a need of products. And if you look even just a recent approval, like isavuconazole, in the US, in the first year of sales, it made more than the last 3 antibacterial products approved. That is dalbavancin, oritavancin, and the Cervexa.
Really?
So,
who won FDA approval for that one?
Sorry? Was Astellas. Basilea Astellas product.
And they made more than like $100 million in the first year?
No, but first year made about $50 million in the US, which again was more than the 3 antibacterials approved recently. So on a business side, I think antifungals remain a great opportunity. The problem is that the development is costly, and we have to make sure we have investors excited about it and willing to invest in our biotech. But I don't want to steal the podium.
I
would like to hear.
You brought up the example of isavuconazole, the Basilea product. So Astellas owns the rights for the U.S., but just last week it was announced that that Basilea sold the European rights to Pfizer for $72 million upfront and $427 million in regulatory and sales milestones, right? And this is for an antifungal for only the European market. So I think it's a testimony to the fact that it's getting the attention of big pharma as well. There are lots of azoles. There's voriconazole on the market, but here's isavuconazole. It's different, but not that different, but it shows that, hey, big pharma is looking at this. Many companies such as ours are looking at this. We see an important commercial opportunity. Another example is posaconazole, right? So that's selling for $600 million annually. It's growing at 30% a year, and it's selling into a generic market. Well, there aren't that many generics, to Marco's point.
The generics that we have tend to be pretty toxic, right?
Well, they're either toxic or there's a high level of resistance. So the market leader, just by scripts, is fluconazole. I think everybody has access to fluconazole. I think Tom can talk about what's driving resistance in India and Pakistan is the use of azoles, right? And this is probably one of the reasons why we're seeing this Candida auris. So it has a lot of drug-drug interactions because it's targeting fungal cytochromes, and humans have cytochromes, and fungi are eukaryotes like us, so there's some crossover activity there. So because these patients are typically immune compromised, they're on a lot of drugs, so having a CYP inhibitor that interacts with other drugs is not a good thing. And so there's a lot of issues with the existing classes. And then there are some toxic drugs such as the poly class, which is the drug of last resort right now.
Is that the amphotericin B?
Yeah.
Yeah, that's a big gun, and it's toxic on the liver, right?
Right.
Yeah.
And still, amphotericin B, despite being despised in some way by anti-infective specialists because, as I was used to say when I was in practice, amphotericin B kills the bugs but kills the patient too. Still, it makes $400 to $500 million in sales every year. So it means that the physicians sometimes just don't have choices. And actually, I think it's interesting this panel where all our 4 companies are really providing something different that can really offer hope to these patients. And is an important point is that the antifungals are difficult to find because the biological machinery of the fungi is so similar, is much more similar, let's say, to mammalian and human cells, that makes it very difficult to find a target that doesn't have enough target effect on human cells.
Well, not only that, but Tom, putting on the scientific hat here for a second, let's assume everybody here is successful. Let's hope they are. Are there some things that you would worry about, like in terms of the antibiotic story, like we've seen with overprescribing, say, leading to more and more resistance, or killing, lots of good fungi? Because as I understand, the vast majority of fungi in the natural environment, we coexist with just fine. There's only, a couple hundred that we have to worry about, right? So.
No, I think that, I'm impressed by all of these companies and others, because there's a few others out there that are in this space. And, for me, again, with my infectious disease hat and also with my public health hat, seeing the problems, I'm excited that we are going to have some new drugs soon and with different mechanisms of actions of some of the other drugs that are out there. Critically important. But at the same time, my public health hat and my antimicrobial resistance hat always says we need to still be good stewards of these drugs. These are miracle drugs, as we know, and as my former director, Tom Frieden, used to say, we don't want to get into this post-antibiotic era where these drugs are no longer effective, and that holds true for fungus as well. We do only have 3 classes. We already know we have isolates at CDC that are resistant to all 3 classes in this Candida auris species, and that's scary. So we need to be thinking about being good stewards, and not just producing more and more drugs, but actually being good stewards with the drugs we have and the drugs that will come out into the market, thinking about vaccine strategies where we can actually not use the drugs all the time, targeting that drug use to the appropriate patients, et cetera. But ultimately, we're about saving lives, and so we need to be able to have these drugs available to us to save those lives from fungal infections.
It seems like rapid diagnosis is a key piece here, because if you have to run a petri dish culture and you don't find out for 3 days, the doctor's kind of got to do something in the meantime.
We're in a new, we're entering a new era, really, in what we call CIDT, culture independent diagnostic tests. And because I'm an infectious disease doc, I love cultures. I like to see the culture. I know it's there. It's approved. Then I can take that culture and I can grow it up against drugs and see if it's resistant. But the bottom line is rapid diagnostics is key for fungal infections. You heard Jeff talk about this 24-hour, 48, 72. Every time you wait another few hours, the death curve just goes up. And so it is critically important to diagnose these things early. They're challenging to diagnose. They unfortunately antigen tests don't necessarily work all that great. So they can be very challenging, but we do need to invest in that space as well. And we're seeing a little bit of investment in that diagnostic space. But certainly, if you can't diagnose the infection, then you're not going to really know how to treat it. So these things really go hand in hand.
Does anybody have a bead on, say, a 24-hour readout, like Cepheid or somebody that does this kind of thing?
T2 is working on it.
Right, yeah, Tom's been working with that company, T2 Biosystems in Boston. That's a rapid diagnostic that I think all of us here are familiar with.
But let me add, from the way we look at it, prevention of invasive candidiasis, getting upstream of the infection, getting upstream of diagnostics, and just say, which patients are at risk? And the risk groups for invasive candidiasis, they're pretty well established. They're patients with central venous catheters, recent surgery, broad spectrum antibiotics that wipe out the bacteria and leave the candida there. So what we would intend to do is to identify patients at risk, vaccinate them before they go into the hospital, before they go into the ICU.
Because
So
we know
to
the
get
longer you stay in the
upstream.
hospital, the greater the chance that something bad will happen
Well,
to you.
if you wait till the diagnosis is done, the death rates go away? Can you get ahead upstream of all that? So that's where the vaccines would fit in. But I think you need all the modalities, vaccines, small molecules, the cloud break kind of technology, you're looking at immunotherapy, monoclonal antibodies, could all be brought to bear.
And what kind of potential market do you see there for this this vaccine, as opposed to, say, the more reactive, let's treat it once we've already identified?
Yeah, well, I mean, you'd have to show that you can identify, let's say, patients that get central venous catheters and maybe had recent surgery, and show that the vaccine is effective in reducing cases of invasive candidiasis. And if you can do that, you could figure out, well, how many people do you have to, for example, vaccinate to prevent a case of invasive candidiasis? So let's say you have to vaccinate 100 patients for each case. Well, a case of invasive candidiasis can be $40,000 in incremental cost. You can get at a purely economic value with that kind of analysis, putting mortality, morbidity aside. But that's kind of, and this is being done in Staph aureus by Pfizer right now, is doing a study in elective surgery for spinal fusion repair. And so they're vaccinating those patients and then trying to reduce the level of Staph aureus. So it's not like a vaccine for everybody, it's very targeted.
Okay, that's bacterial infection,
Yep.
but at least it's one demonstration of this kind of clinical trial design that you
Right.
described.
Correct.
You could do something like that, and you're not going to need to vaccinate 20,000 patients to
No.
get the data that
Right.
you need.
Yeah.
Okay.
In fact, that drug that I mentioned previously, posaconazole, was initially approved for prophylaxis. So it's, a lot of the bulk of those $600 million in annual sales is still from prophylaxis use. It was initially didn't have a treatment label, just had strict prophylaxis. So similar market dynamics
Yeah.
to a vaccine.
I want to make sure people have time to ask questions, and one thing I will say too is that I am cognizant that we are all men up here, and we've talked about some issues that definitely affect women, and I sure hope that the bio-organizers invited some women. I mean, I want to welcome and incorporate that perspective. I just put that out there. So you guys are in R&D. You're trying to explain something to your board, trying to make a case for a program. What are the positives of going to antifungals versus, say, antibiotics? I mean, you've got versatile technologies, can be going in a variety of directions. Are the preclinical models any more predictive here than anywhere else?
Well, certainly for invasive disease, it's highly predictive, as are the models for bacterial infections, invasive bacterial infections. So you do have that benefit, but that's no different from bacterial models. I think it's really what Marco mentioned earlier, is there are fewer companies working in this area. You can argue it's a bigger unmet need, not only because there are fewer companies, but because you have this higher mortality rate. And now you're having these superbug versions of fungi that are now, they're going global. And you have the people sitting up here represent the bulk of the companies working in this area. I see a couple in the audience. That probably constitutes everyone. Right? So it's not as crowded in the field.
Right.
Now, on the other hand, you don't have things like BARDA that can fund, underwrite clinical trials as they do for bacterial products. So if you have a bacterial drug that hits multi-drug resistant gram-negatives as well as some biodefense pathogens, you are eligible to receive BARDA funding that can underwrite a lot of clinical development. And an example of that is Achaogen, which recently reported positive results last year and is gearing up for commercialization. Much of their development was funded from BARDA grants. We don't have access to that as antifungal companies yet. I'm thinking that Candida auris may provide some incentive for BARDA to reconsider that.
Yeah, because the key, again, as we mentioned, is make sure that there is a sense of urgency, in our legislators, in Congress, that they start to put this on the radar screen of the other federal agencies, because BARDA, at the end of the day, their priorities are set by whatever the Congress is really deciding. And so actually we welcome when CDC or even FDA actually they push the point that antifungals should be considered like other invasive, other type of infections.
Well, Department of Defense has a long track record, especially in the antibiotic field, and seems to have paid off pretty well with Achaogen. Their market cap is up. Nobody on this panel is quite yet in the billion-dollar market cap territory. Hope to get there someday. But when you talk with investors, what's the primary pushback?
I'll break in. We're having this conversation as we speak with our board. I'm sure everybody is we've got a portfolio of agents, and for the earlier programs, we're trying to decide what direction to take. And an incremental challenge, I think, in the antifungal space comes down to epidemiology, and Tom's the expert. But we actually built a database from the ground up. What possible indications could one pursue in the antifungal space, regardless of superficial, toenail, mucosal, vaginal infections, invasive? And it ended up to be about 85 different disease entities. The practical reality is, though, you can only develop about 4 or 5 indications because all the others are so rare that you could never pull a regulatory program together. But when you look at what drives billion-dollar sales, fluconazole was 1.6 billion peak. Lamisil, which we won't talk about here today, was 1.2. Vori got to $1 billion. What drives a lot of the sales are these less common illnesses because the docs are using the agents because they're now commercially available. So one of the challenges we've had raising capital is getting people excited about those particular indications that you can actually develop and get to an NDA, because all the others are so rare. They may be more interesting, they may be more... auris, we've talked a lot about that today. It is impossible, and probably will be for the foreseeable future, to pull an NDA together for that indication. So that's an incremental challenge here that I don't think we have in the anti-infective space, is that the bulk of the sales are coming from indications that you really can't pursue from a registration perspective. And so you end up going back, as we did with our lead asset, to more common diseases like nail fungus or RBVC, because they're doable, or try to battle it out in the hospital with IC or IA, where you do have a lot of generic competition and recruitment's very tough, and you guys are in the middle of it. I'm not. That's just something that I think, I'm not sure anybody can do anything about that. But coming back to the GAIN Act, just having that incremental, maybe fast track status for us hasn't been enough to get investors excited about the space. I think we need to have some more dramatic offset of development costs and or payer protection
there are
to
two
get
pieces
beyond
of
these.
legislation that hopefully will be passed this year. One is the one I mentioned previously, is the Disarm Act, which changes the reimbursement paradigm for QIDP products. So one of the challenges for antimicrobials is that they're reimbursed on a DRG code basis, drug-related group code basis, which is capitated. You have an MRSA infection or certain fungal infections, there's a certain payment that you get, right? And so it incents the pharmacy director to use the cheapest drug because they want to keep as much of that payment as possible. Right? So they want to use a cheap drug, and it's probably one of the few therapeutic areas where a physician is going to be limited by the pharmacy director to use a drug that may not be active. You might have 20% resistance in that institution, but you're going to get ceftolozane or something that's probably not going to work. And meanwhile, the patient gets worse as you cycle through these drugs that aren't working until finally, okay, give them the branded. Disarm Act should change that. It provides what's called an NTAP, or New Technology Add-on Payment. So basically, it provides an incentive for hospitals to use a newer branded agent because they know they're not going to lose money because they will get reimbursed from it. So it's more akin to the cost-plus model. In this case, cost plus 0, it's not cost plus 6. That is used for reimbursement for higher price oncology agents or immunotherapies right now.
But is the burden on you to prove superiority in such an instance?
That used to be the case. One of the challenges is that you had to show superiority either in a primary or secondary outcome. So far, there's only one company, and that was Optimer, which now their product Difficid is owned by Merck. They showed superiority in a secondary outcome for Difficid, their C. difficile program. But the NTAP didn't help very much because they were the only company and only drug product that had that. And hospitals didn't want to institute a whole reimbursement program for a single product, right? Now, what under the new proposal under Disarm is if you have QIDP status, you get NTAP on approval. So that should be helpful.
Okay.
The other one is the READY Act, which should be attached to the update of PDUFA this fall. And that is an R&D tax credit. So we're all pre-revenue companies, so which we don't pay taxes on revenues. But basically, it's whatever you have spent in your development program, as long as it wasn't partially underwritten by government grants or contracts. From phase 1 to phase 3, you add all that up. On approval, you get a credit for all that money you invested. And that's a transferable credit, which means, even though you can't benefit from it because you're not paying taxes on revenues, maybe a Pfizer would like to buy that. Well, you can sell that at maybe 10% discount or whatever you negotiate. So these are examples of things that if they pass this year, I think can make a much bigger impact.
It's a game changer.
That game did.
Yeah.
Interesting.
And there are also other options that have been discussed that maybe more difficult, but actually with our group, with Jeff and other, we are thinking, for example, strategies like that helped a lot with tropical diseases. Actually, to have again a lot of drugs developed for tropical diseases was the possibility. Well, this was a legislation that if product was approved for specifically for certain tropical diseases, they will get a priority review voucher that will allow a company actually to accelerate the NDA process of any product, and this could be sold to other companies, and that actually is something that, I found to be personally attractive, also because it will not actually be a burden, let's say, for taxpayers, because
Right.
a READY Act will basically reduce some taxation, but
You don't have that kind of special status here in antifungals.
no, we
or
don't.
antibiotics.
Neither for antifungal nor for antibacterials. But this certainly is something that at a certain point was part of legislation and has been taken out.
I see.
And hopefully it may came back one day when it will be clear that if there is not a strong direct incentive, so the cost of the development is repaid quickly after the product got in the market, I think that one day we can hear again the possibility of priority review voucher.
Right, so someday you could, if you were included on that priority review voucher kind of program, you could be rewarded. You could sell one of these vouchers for a couple $100 million to a company that has some new cholesterol drug. They really, really want it fast on the market. You get rewarded. You still get to be able to sell your own drug if you can get it regulatory approved.
So the question is there ways to potentiate the effect of currently available drugs? Like it happened with antibacterials, when bacteria start to develop beta-lactamases, we developed the beta-lactamases inhibitors, so the antibiotic could return to be effective, could be effective again. For fungi, at least to my knowledge, there are not yet fungi that develop any kind of enzymes that inactivate. So the beta-lactamase approach, I'm not sure will be feasible. But certainly, for example, in azoles, sometimes one of the issues is the development of efflux pumps that really pump out the azoles, and that in that case, a pump inhibitor could be working. And the other big field that I don't think that has been nothing approved formally is actually the possibility of using combination therapies. Is what we do in practice. So when I was in practice, and I would like to know from Tom, is that sometimes to certain patients you are just throwing the sink because you are desperate. So you give an echinocandin, an azole, and also polyens, hoping that something will work out. But there has not been really systematic approach in trying to develop combinations. Of course, combination creates complexity, drug-drug interaction, so you may need to look for products that actually will fit in terms of the ability to be used in combination. But I think that can be another area of development. So the idea of potentiating and using our tools in the best possible way, this can be an option. And Tom, I don't know if you have an option.
The idea of combination therapy, I think is, as you said, in practice, it's probably more of a reality than it is in any kind of indication, because we just don't have studies But I think, for example, if you had a case of mucormycosis, you would probably be treating that patient with 2 drugs up front if you knew it was mucor because of the horrible death rate. That being said, it's another reason for new drugs, new classes, so we can have different combinations. And then to speak of the idea of potentiators, I thought you guys are much more versed in this than I, but I thought there was one company, Harvard or something like that, had a new drug that was sort of a potentiator. It wasn't supposed to be used alone. Sort of like 5-FC, which we use to treat cryptomeningitis. You can't use that drug by itself because the fungal, because especially Candida and crypto, they'll develop resistance rapidly. But with amphotericin, it actually works very, very well in that sort of synergistic approach. And I thought there was a company out there doing that, but maybe I don't know. I'm not familiar with it.
Okay, we have a question here on the side.
Yeah, Marco, you kind of addressed the question that I wanted to ask. Have you guys had any resistance from the FDA or the CDC when you're talking about combination therapies? Because I know multi-drug resistance could potentially increase when you're having multi-drug dosing. Is there any resistance to that, or are they supportive?
I find actually FDA to be very open because, again, very interested at the end of the day is to make sure that new, effective, safe drugs are approved. So they see combination also. The challenge is at the end is always study designs and endpoints. I make a simple example. Aspergillus, clearly there has been some studies reported in the literature where the combination of the quinocandin and azoles have been showing an improvement in terms of efficacy. The issue is that if a primary endpoint for a combination trial remains mortality, well, that can make the study challenging because showing a statistical significant improvement in mortality, which of course will be a requirement for a combination trial, can be quite challenging. So developing secondary endpoints is something that secondary additional endpoints that maybe are secondary today and using them as a primary endpoints can be something that FDA will need to think about and becoming comfortable about it. But also, Tom, to bring you always in the discussion, what will be your opinion?
I mean, the beauty of CDC is we don't regulate anything. So we're out here trying to help set public health priorities and tell folks like you, hey, try to be worried about this disease. And so we are open for all kinds of strategies. We also, on my soapbox, we want to be good stewards of these drugs because we also want them to last. We want to develop new ones. We want those new ones to last. So that's my only caveat. So I would look at things and say, are we being good stewards? Ultimately, are we saving lives as well? And yes, it's a priority to deal with fungal infections. And there's resistance developing. With resistance, actually, sometimes combination therapy is better. It helps because you have a mild level of efflux pumping of one drug, but you add 2 drugs and you overcome that pump, kill the fungus, and they're gone, and you don't actually create more resistance. It's more a lot of times the lower level of drug given for long periods of time, right? And you all know that actually create this. So yeah, the beauty of, I'm all for saving lives, treating patients, prioritizing fungal infections, and I don't regulate anything. So I can say that and feel very comfortable.
Question here in the center.
Where's John Rex when you need him? John sent out an email yesterday. The Europeans, EMA, has just released guidance documents on combination therapy and drug development. I haven't read it, but obviously pertinent to the last question.
So I have some reading to do during my flight back to New Jersey.
Yeah, okay, thank you for that. Next question.
Yeah, Mike Moore from CSA Biotech. We have a small molecule mimic of an antimicrobial peptide that recently was tested at the CDC. We had very, very low MICs against Candida auris. But my question is about the national stockpile. Do you foresee, we have a sister company that just recently got breakthrough status on a coded medical device and hoping to get that into the national stockpile. But our company, CSA Biotech, is working specifically in human pharma. And this is not an area we originally started. We were actually in clinical trials in diabetic wounds, and kind of it morphed into this. Do you anticipate that with the CDC something for in the national stockpile for Candida auris?
You know, I mean, I don't anticipate that in the near future. Yeah, I can't say the extent and the scope of the problem as we proceed. You know, right now we know that thankfully it's incredibly rare. We do, however, know in the places where it has, for lack of a better term, set up shop to a certain extent, that it is being transmitted locally, and it is really hard to get rid of. So, you know, I never say never, never say always in medicine, obviously, but there is no plan currently in discussion of doing anything to the national stockpile for Candida auris. I mean, I think as we think of these MDRO threats, we have to take them seriously, and we have to think about things like that as we're assessing a national strategy.
Is this one of those instances where the sort of lack of hard data on the scope of the problem maybe difficult? I mean, because hospitals are notoriously not very good at reporting about, you know, MRSA or, you know, antibiotic resistance in their setting. You know, it doesn't serve them
I think it's also, I mean, what we've found, at least with Candida auris, is that, you know, it's just really challenging for hospital staff or long-term acute care facility staff to suddenly get worried about a Candida. It's just not something they worry about being transmitted. So we have to get over that culture first to say this Candida is that's why I like to say it's acting like a bacteria. Think of it like a bacteria. So it's somewhat that culture that is actually we're battling with a bit.
And they're not used to drug reps coming around talking about, hey, I got the latest big gun for Canada. You know, you ought to start prescribing that. I mean, it's low awareness, right?
Well, I guess following Tom's point, if we start to see significant number of outbreaks in hospitals, like the one that have been reported in UK, that I think may start to be something that will send a signal that we are really facing a Candida so different from all of other species that have been around today, that will make legislators probably the first to panic. Let's face it, you know, legislators sometimes may need really something that shocks the public, shocks the media, and therefore force them actually to put some thought into it and come in with some legislation. With all the due respect for the legislators.
Scary headlines. Now you're back in my department again. Other questions?
I have a follow-on question for Tom relating to scariness. I mean, fungi have had a lot of different pandemics, but not so much in humans, right? I mean, they've had the Irish potato famine, the Dutch elm disease, chestnut blight, collapse of the bat population, and the 70% of certain populations died out. The collapse of the tree frog population in Central and South America, all due to fungi, which really were quite devastating. So I just wondered, is there any concern that could lead to humans? I mean, bats are mammals. I don't know what caused those and what, you know, why hasn't that happened to humans, or are you worried about it?
I mean, it's not keeping me up at night yet, but I at least will say, someone always asks me, what keeps you up at night? Well, Ebola still keeps me up at night. No. But I don't know if you've ever heard Arturo Casadevall talk.
Oh yeah.
But so you're thinking, so this guy, Casadevall, he's another good fungologist, or whatever you want to call us, who is into fungi, and he blames the extinction of the dinosaurs on fungal disease. And has some pretty good evidence to show that might be true.
Yeah.
And it's climactic in nature because, you know, fungi are the first to grow and die when there's extreme climates. So they're very good environmental indicators about what's going on. As you all know as well, they are the most predominant organisms on the planet. There are more fungi than anything else on this planet. And thankfully, many of them are good.
1.5 million.
Yeah, so many of them are good.
Different species.
A few are bad. Yeah,
according to CDC.
Right, out of 1.5 million species, you know, maybe 100 or 200 are bad so far. But is that one out there, those 1.5 million, that is going to cause serious problems within the human population? I can't speculate. I mean, all I can tell you is that I wasn't anticipating a Candida auris, and yet it's here. And I wasn't anticipating that. I wasn't predicting it, and you know, thankfully I think we can treat it. I can tell you that in some countries, I haven't really talked about the global picture, but in some countries now, Candida auris represents 40 to 50% of their candidemia patients in their ICUs. So that's on the scene from 2009.
Can these hospitals like spray bleach around like
Well,
they
that's
do
what
with?
we're recommending now, because honestly, the holy water of hospitals is quaternary ammonium. That's what we use to clean. Doesn't kill Candida auris. So you have to step it up to a C. diff level disinfectant. And most places aren't just using C. diff disinfectants everywhere, but it's essentially 10% bleach. It's high bleach. So that's what you actually have to do.
Yeah.
And again, but people are like, why do I have to do that about a Candida? It's just hard to process that.
Well, if you're already doing it for C. diff and maybe a couple of other things,
That's
I
agree.
kill the Candida while
because
you're at it.
Candida really is not a Candida patient, is not considered an infectious patient.
Yeah.
It has an infectious disease, but that is not considered, and that is really the big challenge. And the point actually, that may scare. Actually, I never thought about the chestnut blight, thinking, well, what about if it's a human blight, right? And not just the chestnuts. But
Well, the bats scare me because they're mammals, right?
yes, absolutely. And the reality is that these pathogens have been around, billions of years before us, and they will be around billions of years after us. And
Just like the bacteria.
yes.
Okay, I think we could do one more if anybody has one. Good question. Who wants
Yeah, I
to
think
take
one
that
of the
about broad
challenges
spectrum?
there is that you don't know what the species is. So in the U.S., you always talk about Candida albicans, but in fact, in many institutions, it's Candida glabrata, right, which is kind of the predecessor of C. auris that has a haploid genome. It can mutate very rapidly, and so because fungi are difficult to speciate and take a long time, and the importance of having effective therapy very quickly, I think you'd want something that at least covers the genus of Candida.
So actually, my view is that you need both approaches. That is, when you start going back to the point, Jeff, in practice, well, why we start with echinocandin? It's because it covers Candida very well, which is probably 60% of the cases, but it covers Aspergillus too. So you have the time to do additional testing, and of course, if it's a mucormycosis, well, that is start to become a different story, but you start with a broad spectrum. Once you have identified it, well, having actually narrow spectrum, the idea also of monoclonal antibodies, that could be something actually that can make a lot of sense. So I think that there may be different tools for different situations, and having both, I think it will be a significant improvement in our armamentarium.
Great. Well, I think we're out of time, everybody. And now it's time for the cocktail hour. So everybody, please thank our panelists.